So thank you very much for the introduction. Just to reiterate, my name is Daniel Espiritu. I'm a PhD student at Queen's University, and I've been researching the impacts of histone mutations in cancer for the past year. So for some brief background, histones are a group of internuclear proteins that represent the fundamental protein component of chromatin. They can be divided into 5 different types based on their function, but in large, they have the exact same structure. It is composed of 2 disordered tails, the N-terminal tail and the C-terminal tail, and the core domain, which is made up of 3 alpha helices. And by interacting with each other, 2 copies of each of the core histones, being H2A, H2B, H3, and H4, are able to form the histone octamer. And in combination with 147 base pairs of DNA, the histone octamer can form the nucleosome. When with an additional 20 base pairs of DNA and the linker histone H1, they can form the chromatosome. Now, through facilitating interactions between nucleosomes or neighboring nucleosomes, histones are actually able to compact DNA, in addition to being able to modulate some functions involved in DNA, such as DNA expression, replication, and repair. Throughout evolution, histones have been highly conserved. They generally are very similar inside of their structure, at least within the same histone type. But through a series of copy number alteration events, there have been a few instances where histone genes have become duplicated inside the human genome, in addition to slightly varying histones also arising. Although they are broadly similar, they might vary by, say, a couple of amino acids, which can give them a different structure and a slightly different function. So for example, we can look to H2A histones. So if you see here, each dash represents an individual histone isoform, followed by the different genes that actually encode that histone isoform. So the histone gene code is actually very highly redundant, which is kind of acts as a protective mechanism. In case one histone gene is knocked out, then there's always going to be another copy of the gene that can encode for the same protein. And additionally, there are several different variants for histone H2A. Again, they probably perform the same function, although they can vary in certain instances. In addition to having several genes and several different isoforms, histone proteins themselves can actually be modified to perform different functions by the addition or removal of post-translational modification marks. And these actually allow them to interact with DNA and proteins in different manners. So in being able to be post-translationally modified, histones can actually act as kind of a medium for epigenetic modulation of the genome. Now, within the past decade or so, there have been several histone mutations that have been found inside of cancer patients. And some of these have actually been shown to have clear-cut mechanisms of how they actually might promote cancer development. But there have been thousands more that have been observed inside of cancer. And while their implications on how they might affect cancer development aren't quite clear yet, it is clear that they actually occur on highly conserved structures of histones that are important for histone function and consequently may end up causing oncogenesis. So some of the ways that histone mutations can cause oncogenesis is they can occur at those aforementioned post-translational modification sites, leading to alterations in post-translational modification reading, writing, and erasing. They can occur at core histone interfaces. So this can lead to disruption of the nucleosome and its stability and dynamics. They can occur at highly conserved residues that are very important for facilitating interactions between histones and other proteins, leading to altered interactions, which could perturb a number of different functions. They can also occur at the histone DNA interface, potentially again leading to nucleosome destabilization. And they can occur at histone tails. Again, this can vary in effect as to what can happen here, but they can affect protein-protein interactions or protein-DNA interactions. But in large, these mutations are thought to be able to perturb regulation of DNA accessibility, DNA compaction, repair, replication, and gene expression, ultimately leading to oncogenesis. And so, as I mentioned before, there are a few instances where histone mutations have been fairly well documented, but there are thousands more that have been observed inside of cancer. And it is unclear as to what their implications could be inside of cancer patients. So we want to determine which histone mutations are actively promoting cancer development, rather than just being a byproduct of mutational processes underlying within the cells. in addition to determining what mechanisms might allow these histone mutations to drive cancer development. So in order to do this, we've gathered clinical and whole exome sequencing data from 6 different programs and divided them into 2 different data sets. So the first data set consists of patients from the Cancer Genome Atlas, the CPTAC program, HCMI program, MMRF Compass study, and the CMI program. And so from this data set, we have a total of about 12,000 patients with 3,100 histone missense mutations being observed, and about 1 in 5 of every patient having a histone missense mutation. Target, on the other hand, was separated because these are mostly pediatric patients, which may be genetically distinct from their adult counterparts in the other programs. And so from this program, we have about 1,000 patients with 49 different histone missense mutations, and about 1 in every 20 patients having a histone missense mutation. And so here we can kind of just see the number of histone missense mutation observations inside of our integrated data set. So going forward, we're only going to be discussing the integrated data set for today, but we can see the number of observations, observed missense mutations per individual histone isoform. And we do see that this distribution isn't quite equal. There are some isoforms that tend to aggregate mutations more often than other isoforms, but whether this is indicative of the driver mutations isn't quite clear. So we have been using another method to do that. Additionally, you can see here each of the individual histone types, being H4, H3, H2B, and H2A, H1 have all been color coded. And as I mentioned, although we are focusing on missense mutations, there are several other different histone mutations that have been observed inside our data set. Although missense mutations do represent the largest single variant classification for a mutation out of all possible variant classifications for mutations. So in order to quantify or predict which mutations are actually acting as drivers rather than just passenger mutations or mutations that don't actually actively contribute to oncogenesis, we've been using MUTAGENE. We opted to use MUTAGENE over other methods, such as, say, frequency, because methods such as frequency kind of tend to misclassify mutations that are observed at the extremes. So say if we had a mutation that is observed at a low frequency, and if we were to classify that as being either a passenger or driver just purely off of using frequency, we might say it is a passenger. But it is possible that that mutation, although not observed that often inside of a given cancer type, is actually a driver. It's just a very rare driver. And conversely, it is also possible that a mutation might be observed several times inside of a given cancer type, but it actually may not be a driver. It may just be the passive byproduct of a mutational process that is actually affecting patients inside of that cancer cohort. So without getting into the MUTAGENE method too much, it gives predictions as to whether a mutation is a driver or not for individual cancer types. And it does this by calculating expected mutation frequency, or the expected number of times that mutations can be observed inside of a given set of patients for a given cancer type. And then it takes the observed frequency of that mutation and normalizes it by the expected frequency in order to get a continuous scoring metric, for which thresholds can be applied to determine whether that mutation is a driver, potential driver, or passenger mutation. And so from our integrated data set of about 3,000 missense mutations, we have predicted about 322 of those histone missense mutations to actually be drivers within the respective cancer types. Additionally, in comparing frequency and our predictions for MUTAGENE, we can see here just on the left, this is the total number of histone missense mutations inside of our data set partitioned by the individual histone type. And then here we can see histone missense mutations, which have been predicted as drivers inside of our Pancancer cancer model. And so these are just restricted to driver mutations. And you can see that when comparing just all mutations by histone type to driver mutations by histone type, that the proportions actually do change quite a bit. And so while MUTAGENE is able to tell us whether or not a histone mutation might be a driver or not, it doesn't really give us many insights into possible mechanisms that actually can allow this mutation to actively drive oncogenesis. And so in order to kind of begin investigating possible mechanisms, we've been using histone interaction networks or histone interactomes. So these were created by our collaborator, Dr. Yonghui Peng, using structural data from the protein data bank and cross-linking mass spectrometry data. And they detail interactions between histones and other biomolecules, other biomolecules being DNA, interactions between histones and other histones, and interactions between histones and other proteins, such as, say, DNA repair proteins or DNA replication proteins. And these are individual residue level interactions. And so by combining these with our predictions for MUTAGENE, we've been able to see that about 58% of the mutations we predicted as drivers overlap with an interface. So we have about 44 overlapping histone DNA interfaces, 136 overlapping at histone-histone interfaces, and 108 overlapping at histone other interfaces, other just being kind of a category that includes any biomolecule that is not DNA or another histone protein. And so while histone interaction networks are very useful for determining, if a mutation could potentially disrupt an interaction interface, it doesn't actually tell us the degree to which it might actually interrupt these interfaces. So for example, from the interactome, from MUTAGENE, we know that this mutation, H3.1 E105K, has been predicted as a driver inside of our pan-cancer model. And it overlaps with histone chaperone interfaces. So for example, here we can see the ASF1 histone chaperone with yellow coloring the H3.1 E105 residue. And here we can also see the FACT complex subunits, SPT16 and SSRP1, again with E105 being highlighted inside of yellow. And so we want to know if the E105K mutation can potentially disrupt histone chaperone function through perturbing interactions between these various proteins. So in order to do this, we've been using MUTABIND2, which is able to predict mutation effect on binding affinity, represented in kilocalories a mole as delta delta G. And so when we actually use MUTABIND2 to quantify how the E105K mutation might disrupt these interactions, we see that using a threshold of about 1.5 kilocalories per mole of an increase inside of binding affinity, we see that the E105K mutation might not interrupt the interaction between ASF1 and H3.1 or SSRP and H3.1, but it could potentially disrupt the interaction between H3.1 and SPT16, possibly leading to disruption inside of the FACT complex being able to transport the H3.1 histone and incorporate it inside of nucleosomes. So overall, from this project, what we've been trying to do is use whole exome sequencing in addition to various driver quantification methods to identify candidate driver mutations, histone mutations specifically. For example, those H3.1 E105K mutations that we discussed, in addition to finding general mechanisms whereby histone mutations can actually possibly promote oncogenesis. And with that, I'd like to thank my supervisor, Dr. Anna Panchenko, and our collaborators, Dr. Yonghui Peng and Dr. David Lansman, the Ontario Institute for Cancer Research for this platform today, NSERC, all the patients and the organizers of the programs that we've been using to gather clinical data and whole exome sequencing data, in addition to the National Institutes of Health. So thank you very much, and I think we have some time for questions.
Okay, thank you, Michelle, again for the introduction and arranging this presentation. I really appreciate it. As the title of my talk suggests, today I'll be presenting some of our work, which I basically did back at Yale to build methods to interpret genomic variations in cancer. So this will be the quick outline of my presentation. I'll be giving a brief overview of cancer genomics, particularly talking about how people think regarding identifying drivers from so-called passengers in cancer genome. And one of the approaches you can apply to identify these drivers are something called quantifying their molecular impact. So I'll be talking about some methods which we developed over the years to quantify molecular effect of different kind of genomic variations. And then in the second half of my presentation, I'll focus on one of the study where we looked especially for the passenger landscape or so-called non-driver landscape in thousands of cancer genomes. And then toward the end, I'll talk about some of the future research direction which my lab is taking here at Princess Margaret. So as most of you already know, that there are multiple hallmarks which are often associated with the typical tumor cell, and majority of these hallmarks to a certain extent can be related back to the presence of genomic instability and somatic mutations in cancer genome. And that's one of the reasons why over the years there has been a lot of interest in generating genomic profile for cancer patients. And what has given rise to the field of cancer genomics is the sharp decline in sequencing cost, which allowed consortia efforts such as ICGC and TCGA to generate genomic profile for tens of thousands of cancer patients. So for a given cancer patient, we can sequence their tumor DNA as well as adjacent normal DNA from the same tissue for the patient. And comparison between tumor and normal gives us insight into somatic genomic variation profile, whereas a comparison between normal DNA with respect to reference genome gives us insight into what we call germline genomic variation profile of the patient. So compared to the disease, cancer is a challenging disease to study because of the underlying issue of genetic heterogeneity, which simply means that when you look at genomic profile for two cancer patients, either within a cancer type or between different cancer types, they're quite distinct. And it makes it really challenging when it comes to identifying these key or actionable mutations, which we like to identify and target therapeutically. So the field of cancer genomics, we call these actionable or key mutations as driver mutations, which are supposed to provide selective growth advantage to tumor cell. They're typically handful in number. So depending upon cancer type, you will observe anywhere between 2 to 8 driver mutations in a given cancer patient. And the genomic elements where these driver mutations reside, the so-called driver elements, are generally identified by searching for what we call signal of positive selection. So simply put, what that means is that we are looking for gene or genomic elements in cancer genome which are more frequently mutated compared to random expectation. So in the canonical model of classifying cancer mutations, we have a handful of driver mutations, and the remaining thousands of mutations are considered to be passengers, which are supposed to be inconsequential for tumor development. So now you can understand there's a challenge. How can we identify these handful of drivers from these thousands of so-called passengers? So over the years, multiple methods have been developed to identify these primitive drivers. And broadly speaking, these methods can be classified into 3 distinct categories. First is what we call recurrence-based method, where you're simply looking for genomic elements or genes in cancer genome which are more frequently mutated compared to a basal background mutation rate, which you should observe in a typical cancer genome. In functional impact-based approach, along with the frequency of mutation, you also consider the molecular effect of each cancer mutations to identify genomic elements which are significantly enriched for high molecular impact cancer mutations. And finally, in clustering-based approach, your goal is to identify sub-regions either on the genome level or on the encoded protein sequence or structure level, where you see significant enrichment of cluster of cancer mutations. So in today's presentation, I'm going to focus mostly on functional impact-based approach. How do we identify or quantify these functional impact? So, and the way you quantify these molecular functional impact varies based on whether the mutation is coding or non-coding. So in case of coding mutations, you can integrate protein structure with cancer genomics data to assign molecular impact score to them, especially in the case of non-synonymous mutation. And this integration approach is very powerful because it gives you mechanistic insight into how these mutations might be influencing protein stability, which in turn influences protein functionality, which ultimately is driving cancer progression. And if you integrate this mechanistic insight with clinical data or drug interaction databases, you have potentially opportunities to come up with new therapeutic avenues to treat cancer. So that's why over the years, there has been a lot of interest in integrating protein structure data with cancer genomics data. And majority of these methods have tried to understand influence of these, especially missense mutations on the global stability of protein structure. But the problem with these non-synonymous mutations are that their impact on the global stability of protein structure is very subtle. So instead of looking at the influence of these missense mutations on the global stability, we decided to quantify the local perturbation they might be inducing. And this idea we borrowed from previous protein folding studies, which argues that when you look at the native structure of protein, majority of its residues are what we call minimally frustrated. So simply put, if you take a native structure of protein, suppose you have this toy example over here. On the native structure of protein, you define a local neighborhood based on certain distance threshold. You'll observe that in this local neighborhood, in this toy example, an asparagine residue is surrounded by negatively charged amino acid residues. So in this local neighborhood, asparagine is experiencing favorable interaction, so it will be what we call minimally frustrated. However, if you mutate this asparagine to an aspartate, which is a negatively charged amino acid residue, in this negatively charged local neighborhood, now it will experience unfavorable interaction or will become maximally frustrated. So our goal is to quantify this change in frustration profile and use that as a metric of molecular impact score for cancer missense mutations. So long story short, when we applied this on mutation data, non-synonymous mutation data from TCGA, we found distinct pattern for oncogenes compared to tumor suppressor genes. So in case of oncogenes, what we found was that surface residues tend to be more locally perturbed compared to core residues because of these missense mutations. Whereas in case of tumor suppressor genes, core residues are getting more locally perturbed. And one way to explain this observation is that in case of oncogenes, because the surface residues are getting more locally perturbed, in order to quench for that extra frustration, the mutated oncogene are likely to interact with non-specific binding partner, which might be leading to gain of function effects, which are often associated with oncogenes. Whereas in case of tumor suppressor genes, because the core residues are getting more locally perturbed, that perturbation will transmit throughout the protein structure, leading to global destabilization, which might be leading to loss of function effect. So you can see from this example, not only can quantify molecular effect of these missense mutation using this approach, integration of protein structure also gives you mechanistic insight into how they might be influencing protein functionality. But this approach is useful to quantify molecular impact score, but we also wanted to identify putative cancer driver genes. So one of the way you can identify putative cancer driver genes by integrating protein structure is by looking for these cluster of mutation, which I was alluding to in my introductory slide. And people have done that where they have looked for this cluster of mutations on the static structure of protein. But we know from protein biophysics that in order to function properly, protein needs to move around. There has to be dynamics element. So we hypothesized that if we integrate protein motion on top of protein structure, maybe we'll be able to identify additional putative cancer drivers, which we are missing just because we are just looking at the static structure of protein. So for this, we developed this framework where for a given gene, if we have encoded protein structure data available, we convert that protein structure into form of what we call residue interaction network. So in this network, each node corresponds to a residue, and the connectivity between two residues signifies that those two residues are close together on the protein structure. Now, simultaneously, we perform coarse grain simulation to identify residues which have correlated or anti-correlated motion. And based on the extent of correlation or anti-correlation, we assign weights to each of the edges in this residue interaction network. Now, once we have this weighted residue interaction network, we perform a simple network partitioning to identify what we call residue community, such that each residue community is only occupied by those amino acid residues that have correlated motion with respect to each other. And then afterwards, we map cancer missense mutation data onto all these communities and identify those communities which are significantly enriched for cancer missense mutations. And any gene which encodes for protein structure, which has at least one mutational hotspot community, we consider them to be putative cancer drivers. So we applied this framework using TCGA PanCan Atlas dataset, especially looking at missense mutation, and we ended up identifying 434 putative cancer driver genes. Many of them were well-known cancer driver genes, but we ended up identifying additional putative cancer driver genes, many of which were differentially expressed among tumor samples, as well as were enriched in different cancer-related pathways, signifying that a large proportion of these additional driver might be playing important role in cancer. So far, I mostly focused on coding mutations, but the overwhelming majority of cancer mutations fall in non-coding region. And quantifying their molecular impact is not straightforward. So as part of the PCAWG project, which OICR was one of the leading organizations to work on, I also had an opportunity to look at whole genome sequencing data. And one of the things which we did as part of the PCAWG project was to quantify molecular impact of both coding and non-coding mutation. And as I was saying earlier, quantifying the molecular impact of non-coding mutation is not very straightforward. But one of the places where you can quantify molecular impact of non-coding mutation are those regulatory regions which encompass of these transcription factor binding site. So when you have mutation in a transcription factor binding site, you can either create a new binding motif leading to what we call gain of motif event, or destroy an existing binding motif leading to what we call loss of motif event. So we simply ask this question: when we look at binding site for different transcription factors across genome for different cancer types, do we see enrichment or depletion of these gain or loss of motif event? And indeed, we found that there were certain transcription factors such as PRRX2, GATA, SOX10, and so on, which were enriched for gain of motif event. And then there were certain transcription factor binding site where we saw loss of motif event enrichment, such as EP300, TF-AP2, and so on. And although I'm not showing you data today, but when we looked at the downstream gene, which were regulated by these binding sites, we found that indeed the enrichment or depletion of these gain or loss of motif event correlated with the differential gene expression, suggesting that a subset of them might be leading to functional effect. So beyond point mutation, though, there are also other kind of genomic variations which you observe in a cancer genome, such as what we call structural variations. So operationally, you define structural variations as variants that have length greater than 50 base pair, and they can go up to mega base pair, and in certain cases, chromosomal arm length deletion and duplications too. And because they are larger length size, these structural variants end up affecting larger proportion of genome. And when you count the total number of nucleotides which are being affected by structural variations, they are much more larger compared to SNVs and indels combined. So suggesting that these structural variants might have important role to play in disease. Also, because they're larger length, they can end up affecting both coding as well as regulatory regions simultaneously. Another thing which I wanted to mention was that these structural variants can be further subclassified based on how they end up affecting DNA segments in the genome, such as deletions, duplications, and so on. So despite the important role in cancer and the diseases, their lack of tools and methods to interpret these structural variation. One reason is that compared to point mutations and to a certain extent indels, small indels, we do not have accurate tools to identify structural variation as accurately as we can do for smaller variants. Another aspect to consider is that because we cannot identify them accurately, there's not too much development toward interpreting them that much. So to address this challenge, we ended up developing a machine learning framework, which basically look at structural variation for a disease set and the corresponding control set. And you can do this for somatic setting as well as germline setting, and look at various underlying genomic and epigenomic features. And when you're looking at epigenomic or functional genomics features, you want it to be tissue specific because tissue specificity matter a lot when it comes to non-coding genome. So in this framework, we generate this complex feature landscape, and then our goal is to assign a score which reflects how likely a given input SV is close to well-known cancer-related SV compared to a benign set of SV. And based on that score, we prioritize or assign what we call pathogenicity score to structural variation. And when we looked at different cancer cohort, again from PCAWG, we found that our method was doing pretty good job, not as great as we would like, but still it was a good amount, decent amount of job. This is rock curves which I'm showing for 6 different cancer cohort which we looked at. We also looked at the downstream genes which were being affected by especially the high impact SVs, and we found them to be relevant for a lot of cancer related pathways. I'm not showing the result, but we also found our impact score aligned well with the recurrence-based driver SV identification. So suggesting that our method is working quite well. Now, so far I focused mostly on the driver aspect, but we know that drivers are handful in number, and the overwhelming majority are classified as non-drivers or so-called passengers. So we were also interested in how does these non-driver play a role in cancer? Are they really inconsequential, or they might have a role to play? So for this, we came up with this conceptual idea that the canonical dichotomy between driver and passenger is of convenience, and maybe we can extend this into an extended model where, along with standard drivers and neutral passengers, we can have additional categories of mutation, such as undiscovered drivers or deleterious passengers. So undiscovered drivers are those non-driver mutations which have been incorrectly labeled as passengers, but in reality, they are promoting tumor growth. So they are providing positive fitness effect to tumor cells. On the other hand, deleterious passengers correspond to those putative or non-driver mutations that have negative fitness effect on tumor growth. So they are inhibiting tumor progression. And you can further subclassify these undiscovered drivers or deleterious passengers based on the extent of positive or negative fitness effect which they're imparting to tumor cell. So for example, you can have strong undiscovered drivers, which are basically rare cancer drivers, which your recurrence-based method is likely to consider as passengers just because you do not satisfy the statistical power criteria, which you don't have enough sample to identify them. On the other hand, weak undiscovered drivers are those non-driver mutations which work together either through epistatic interactions or through additive effects to drive cancer progression. And similarly, you can further subclassify deleterious passengers as strong or weak, depending upon the extent of negative fitness effect which you are imparting to tumor cells. So with this extended model in our mind, we looked at a different aspect of non-driver mutation. And one of the snippets or the one set of analysis which I wanted to share with you today was this additive effects approach. So if in the GWAS germline community, people have shown that when you look at handful of mutations falling in the GWAS loci and use those handful of mutations to estimate heritability of a complex trait such as height, you do a very poor job in estimating the heritability of height as a trait. However, when you start aggregating common mutation and rare mutation, you are able to recover back the heritability estimate of height as a trait. So with this idea, we borrowed this idea in the context of somatic setting and asked, does the aggregate effect of these non-drivers of putative passenger mutation can help us predict phenotype of a cancer? And if it does, how much additional predictability does it provide? So for this, again, we borrowed the idea of additive effects model from germline studies, where additive effects model is simply aggregation of genotype of each mutation along with their effect size, along with a residual environment term to predict phenotype of a given sample. So in our case, our phenotype is binary, 1 meaning cancer, 0 meaning non-cancer. So we came up with this setting where for each cancer genome, we left out driver mutations. And for each non-driver mutation, we generated a corresponding null mutation or random mutation by shuffling that mutation while considering their underlying genomic covariates and the trinucleotide context, which is very important because you want to model the background or mutational processes as closely as you can. So now using this randomization approach, we were able to generate this case control kind of setting where case corresponding to cancer genomes and control corresponding to these non-cancer genome. We converted this into form of genotype matrix, but did the normalization and took the effect size of mutation into account to predict phenotype of sample being cancerous or non-cancerous. Now, our goal is to quantify the aggregate effect of these putative passenger mutations toward the observed phenotypic variance which you predict using this model. Then we made our model slightly complicated to stratify it such that you can contribute the contribution of coding mutations, coding drivers, and other non-driver mutations such as coding promoters and other non-coding non-driver mutation. And when we applied this model into for eight different cancer cohorts as well as on pan-cancer level, we found that obviously driver mutations has the highest predictability to identify sample as cancerous versus non-cancerous. However, when you add aggregate effect of putative passenger mutations on top of driver mutation, the predictability enhances significantly, suggesting that indeed maybe some of these non-driver mutations which we are considering passengers and ignoring them, some of them might have a role to play in cancer. And then our stratified model also showed that, long story short, is that coding non-driver mutations has the most contribution toward predictability of our model, followed by promoters and other non-coding mutation, which again makes sense because you'd expect that coding mutation will have larger functional impact, and that's why they have larger say in predicting the phenotype. So with that, I'd like to just conclude and talk about some of the future research directions of my lab here at Preem. So in conclusion, I assure you that we have developed over the years methods to interpret molecular effect of coding as well as non-coding mutations and genomic structural variations. So going forward here at Princess Margaret, I would like to continue some of the work, build upon some of these works. So I'm trying to build methods related to clinical data and try to understand how does these mutations have effect on drug binding affinity as well as efficacy of drug. There's a lot of work needs to be done in the non-coding space, trying to build accurate model to identify underlying functional motive of these non-coding regions and understand how these non-coding mutations might be playing a role in cancer progression. I'm also interested in building methods to identify these structural variations accurately, which I was telling you earlier that it's a little bit challenging, but with long-read sequencing data, it's possible to do a much better job. So I'm interested in building those kind of methods. And one of the interpretation aspect which we can look at as far as structural variation is concerned is understanding their influence on 3D genome and how it might be influencing gene regulation. And finally, I'd like to build upon this idea of additive effects model to understand in general how does cooperativity works in the context of cancer, either looking at the genetic level or in the context of cellular level cooperativity, and how does that cooperativity changes as tumor progresses from a primary to a metastatic stage cancer. So with that, I just want to acknowledge a lot of people who worked on this project. Obviously, all these projects were done back at Yale. So I'd like to thank Mark Gerstein, who was my PI at Yale, along with various lab members and PCAWG consortia. Through these interactions, I was able to do all these works. So thanks to all of them. And thank you for listening, and I'll be happy to take any questions.